Yeah, thanks very much, everyone. Good morning, and I wish I could deliver this talk in French, but thanks very much for accepting that to be in English. So it's a real pleasure being here and be able to present some of our findings in Trinity College Dublin here. I would like to thank Polio-France, Mr. Robert Cordier, and all of the organizers, and especially for the invitation. So I'm going to talk about the network level dysfunction in polio, and more specifically, I will present our findings from a corticomuscular coherence study, which seems to be a jargon, but I try to make it much simpler. So briefly, I'm going to introduce our team and the research challenges we face in neurological and neurodegenerative conditions in managing them, treating them. And then I focus on a specific study that should be of our interest, and then some discussions and summary of this finding and the relevant findings. We are a team based in Trinity Biomedical Sciences Institute, which is in Dublin city center, definitely far from where we are now in France. And we have links with the National Polio and ALS clinics in Beaumont Hospital, and we have links with research facilities in clinical research facility at St. James's Hospital. So we are a large team of researchers, 16 people, 3 principal investigators, and so people have different expertise ranging from clinical neurology, neuroscience, engineering, just like myself, psychologists, biologists, physicists. We are a diverse team from 11 nationalities. Now, so within our team, I'm responsible for a smaller team that focuses on signal analysis of the signals from the brain and from the muscles. More specifically, people that have contributed to this work include current and previous team members, Dr. Amina Kafi, Mr. Saroj Bista, Professor Orla Hardiman, and all of our collaborators and assistants. So so about the research challenge. So I'm sure you are already very aware of the challenges we face in managing any neurological condition. But so we all remember the situation where a family member or just ourselves, discovers a new pain, a new tremor, a new weakness in some part of the body, and then what we end up thinking. So is there a cure for that? So what is it? Can it be diagnosed? And if it's diagnosed and if it's manageable, the treatment and rehabilitation or any intervention, does it have any effect? How can we measure the potential effect of intervention and improvements? So I'm talking about drugs here more specifically. So that's the case in neurological conditions. But in polio, similarly for any physical or neural rehabilitation, that still applies. So we lack biomarkers for quantitative measurement of improvement. And by that improvement, I mean the function in the nervous system. What we need to measure, knowing the fact that the diseases can be very, very heterogeneous, are biomarkers that specify what happens in different networks of the nervous system, and that's what we need to quantify. Now, so let's see a very simple but basic, but admittedly very simplified version of the neural circuits in a human body. On the right, you see the networks, the sensory motor networks, including the upper motor neurons in the brain and the lower motor neurons in spinal cord, and the sensory pathways that contribute to generation of movement and muscle activity. Now, what happens if these pathways degenerate? So the most common types of degeneration, I have listed a few of them. Either both the upper and lower motor neurons in both the brain and spinal cord degenerate, such as ALS, or only those motor neurons in the brain degenerate, such as PLS, primary lateral sclerosis, or those in spinal cord. And that includes neurodegenerative diseases such as primary progressive muscular atrophy, PMA, spinal muscular atrophy, and in addition to these neurodegenerative conditions, post-polio syndrome caused following poliomyelitis infection. So I will interchangeably use the terms because we all know so what the exact definitions are. So what matters here is that polio is a lower motor neuron dysfunction from a scientific perspective. Now, so what's the potential effect of dysfunction in lower motor neurons in spinal cord in polio, and the effects that they exert in broader neural systems in the spinal cord and in the brain, so which may not be adequately discussed to date. In other conditions such as neurodegeneration, more specifically ALS and MND, motor neuron diseases, so there are extensive attempts to quantify how much the lower motor neurons in spinal cord are damaged. But what is probably missing a little bit is the effect of these dysfunctions in spinal cord, so on the network level function. So, and that's probably what we need to investigate further. In our team, so we focus on neuroelectric biomarkers, and by that we mean neural signals reflecting neural activity of the brain and of the muscle. And so such measurements provide benefits for us because they are non-invasive, inexpensive, and they have the potential to reflect what happens not only at a focal level, say in the brain or spinal cord, but at a network level. So we will see that. We use series of experimental techniques, data analytics, and experimental paradigms, and such as EEG, EMG, and different types of analysis. I will focus today in this talk on only a few of these methods, more specifically EEG that we record from the brain over scalp, EMG from over muscles, and then so how they reflect the network function during a motor task. So that's the study that I want to explain today, and I hope the results are of some interest to you. The study done by Dr. Coffey, Mr. Bista, and my colleagues. And so we want to see how polio affects the supraspinal motor networks in nervous system, something that may not be very trivial given the fact that the damage is in spinal cord. Now, let me introduce briefly to the methods that we used for this study. We use high-density electroencephalography. So these are the recorded signals from the brain. And what we do, in addition to just recording them, is a spectral analysis. And that simply means we decompose and separately analyze the fluctuations and waves, the neural oscillations in different frequencies. That means slow oscillations, medium speed oscillations, fast oscillations, to see what happens in the brain. We do the same for what we record from the muscles, the neural activity manifested as contractions in muscles. And again, we see how neural oscillations are reflected in muscle function. So in order to see what brain regions and how much they contribute to the control of a movement or a motor task, so we see the synchrony or functional connectivity between the oscillations in the brain and in the muscle. And that's the phenomenon referring to that. Corticomuscular coherence is a synchrony between cortical activity or also subcortical activity and the muscle. And so we studied this synchrony in a group of people with polio and a group of people without polio. 24 people with polio and 11 people without polio, which were also age matched. So, when the participants of our study exerted this force, a pincer grip, so we measured the synchrony between EEG and EMG. And what we typically expect when people don't have polio in such conditions is that the area just over the primary motor cortex responsible for the activation of the muscle shows a typical synchrony at frequencies about 20 hertz. So the axis here, frequency, you see here the blue area shows what we expect to see, and the higher the values, it means there is higher level of synchrony between the muscle and cortex. So that's what we expect to see in healthy individuals, but what happens in people who have experienced polio? Do we see the same thing, or we see something else that we don't expect? Yes, that's what we want to understand. So what additional phenomena we observe? Now, let's see what we found. Interestingly, so we did not see that typical beta band activity pattern at about 20 hertz. Instead, we saw a communication between the cortex and muscles at much higher frequency, faster oscillations. So instead of 20, about roughly speaking 40 hertz. And so not only we saw that, but we saw that in regions that are not primarily associated with the control of the muscle. So not necessarily primary motor cortex, but frontal and parietal part of the cortical regions. So that's an abnormal communication between the brain and muscle that we observed very consistently. So of course we saw additional unusual patterns of communication as well. That on its own, so and in addition to that, so when so our researchers compared the communication between those individuals who had polio in infancy as opposed to later childhood, we saw the same patterns in both groups, but with some differential patterns that slightly distinguishes them. So, so what we might infer from these findings is that additional brain regions and networks start to take part in the control of movement in people who have had polio in the past. So, and this is probably due to the impairment of the main pathways at the lower motor neuron level in spinal cord that leads to additional engagement of the cortical regions. So what's the exact nature of these changes, whether they are adaptations, compensations, neural plasticity that needs further investigations. But what probably helps us to study this phenomenon further is looking at this type of corticomuscular coherence in neural conditions who share some aspects with polio. And one of them is spinal muscular atrophy, in which the same lower motor neurons in spinal cord degenerate. And interestingly, so even though we studied that in 6 individuals, we see that the same gamma band corticomuscular coherence, again at the unusual frequency of 40 hertz, show up. And again, they appear in regions that are not primarily motor areas, but, for example, frontal areas. So that's a good similarity in what we found in lower motor neuron dysfunction. So the similarities in the two conditions imply that the abnormal corticomuscular coherence patterns originate from the disruptions in motor circuits due to the dysfunction at spinal level. What we further studied was to see what happens if people who have damaged upper motor neuron in the cortex. So we studied the same phenomenon in these groups. We did see some abnormal patterns again, but not exactly in the way that we observed in the dysfunction of lower motor neurons in spinal cord. That included abnormal communication at different speeds of communication in the motor area as we expected, and in the other side of the cortex that is not typically an active contributor to movements. So these changes correlated with the clinical measurements found by neurologists, so confirming that they are definitely related to the disease. And importantly, when we look at the map that which brain regions have relationships with such clinical impairment, we see that those negative correlations in the frontal areas show that when there is higher impairment, the contribution of these regions to movement goes down. Instead, the other regions shown in red start to contribute more to the control of the movement, so in presence of upper motor neuron dysfunction. So these findings are encouraging, but whether they are characteristic or not, so that's what we need to find out. If they happen only, for example, in polio or lower motor neuron dysfunction of spinal motor neurons, so whether we can dissociate different types of the disease accurately, how much discrimination we can get so that we can use them as biomarkers when we want to measure the efficacy of treatments, that is yet to be understood. Of course, we plan to project these abnormal patterns to the underlying brain regions to more accurately figure out which exact brain regions contribute to this abnormal network level community. So what we expect, hopefully, in the future is something like this. So we did this study recently in ALS patients, and then we measured the activity of networks in the brain, and that could successfully identify subgroups in the patients that we couldn't identify from just clinical examinations or other clinical tests. So if we can move to this direction, not only for ALS, but for any neurological condition, so we hope that we can find measurements that tell what happens in different parts of the nervous system and how much. And just to summarize, corticomuscular coherence indicates altered supraspinal motor networks in polio, and of course in other cognate neurological conditions. So the benefit for drug discovery is that so we can potentially find biomarkers that reflect the not only focal but also network level dysfunction, and then we can hopefully measure what happens right or wrong in clinical trials, interventions, rehabilitation. And of course, we may have new tools in the future. I would like to thank everyone who contributed to this study, and also especially participants, people with post-polio syndrome, PLS, and other conditions, and the funding bodies and everyone involved. And thanks very much for listening to me in English. Really appreciate that. And so if there are any questions, I'm more than happy to talk about it more.
Well, thank you very much, Bahman, for this very interesting talk. Very interesting. Thank you very much. Est-ce qu'il y a une ou deux questions?
Do we have any questions?
Alors, il y en avait une là-bas, On va aller très vite.
We have a question over there. Quite quickly, please.
Il vous faut le micro.
Sorry, thank you. I thought that was really interesting. Have you ever seen, when you do these tests, where the oscillations have somehow decreased more from the 40 to the 20? Is there any signs that they can be improved in future?
I'm not sure if I got the question right. So you mean the change in the frequencies or the intensity of frequencies?
Sorry, I didn't hear that.
So I couldn't get your question. So do you mean the change in the frequencies or change in the intensity of oscillations?
So have you ever seen cases where you can do interventions to get that improvement?
Yeah, so no, we haven't done that yet. So what we ideally hope to see is whether any intervention, treatment, or therapy can potentially alter these measurements. That's what we hope to be the case in the future. And if we can correctly reflect what's happening at network level, that's very promising. But for now, so these are just interesting findings to monitor.
Thank you. Thank you.
Thanks.
Excusez-moi, est-ce que vous avez entendu?
Have you heard about the digital bridge that allows, through thoughts, to stimulate muscles and allow for some disabled people to walk again? It's been mentioned in the news for 2 days.
Sauf erreur, vous abordez la question.
I think you're talking about communication through an interface, a brain-muscle interface.
On peut en parler après, hein, mais c'est un autre.
I'm sorry, it's another topic.
Et puis.
Bonjour, monsieur. Je voudrais savoir si.
Good morning. I would like to know if there's been some progress in diseases such as epilepsy. For the treatment, I mean. For epilepsy treatment, have you seen any progress?
Translation.
oui. No. ببخشید. رضا هستم. به فارسی میگن که آیا برای اپیلپسی درمان پیشرفت کرده یا نه؟
Well, epilepsy is not my expertise. Although similar techniques in terms of electroencephalography is used, so I'm not directly involved in that. So I have colleagues who work actively on epilepsy, but that's not really my expertise. So from a technical perspective, very similar methods are being used. Yeah.
Excusez-moi, la douleur sur un membre.
I have a question. The pain on a limb that has been affected by polio is higher than on a normal limb. Can this be assessed by your study?
Pain in polio with your works. The difference of pains in a paralyzed member and non-paralyzed member.
Yeah, so pain is a slightly different aspect. So here we studied how the neurophysiology, the activity in the nervous system is related to mechanical generation of force, whether or not with or without force, without pain. So some of our participants were experiencing pain while some others were not. So that's not conclusive for pain, but something that I can add is that I work with a colleague on patients with polio, in which my colleague measured the effect of polio on the motor unit numbers estimated in different muscles. And so then there was just a trend, not a meaningful difference in people who had experienced pain or not. So what I can say is potentially pain has some relationship with the motor unit remodeling in muscles, but so whether our findings can reflect that, so we don't know.
Okay, thank you very much, Bahman. We'll have to move on. Thank you very much for your talk, very
Thanks,
interesting.
everyone.